This video on RNA Expression Vector is broadly split into three major parts: why we use them, some rules and considerations around their design, and finally a couple of examples to help guide your thought process when it comes to RNA expression vectors. Typically, when we talk about molecular expression of a gene or a genome, we refer to the DNA template being converted into RNA and maybe a protein. These are the processes of transcription and translation. In this chain of information transfer, you notice that there are two possible outcomes. For the purpose of this video, we assume that the chain of outcomes stops at the RNA level, and therefore our concern is with vectors used to make only the RNA. This also means that the protein outcome is often not desired from these vectors. Another way of saying this is that the RNA is non-coding in function, and that is desired. So, why do we do this? Back in the day, for DNA sequencing, you needed single-stranded RNA, and phage expression vectors were used to make RNA. Today, if you do hybridization experiments like fish, you need RNA probes. Studies around RNA tracking and localization need single-stranded RNA. There is evidence to suggest that localization of RNA can affect cellular physiology. If you do RNA interference for knockdowns, you need double-stranded RNA or hairpin RNA. The guide RNA used in CRISPR is a single-stranded RNA as well. Oftentimes, RNA vaccines or treatments use RNA as a product. Among many other applications, I hope you can see why RNA expression vectors are important. In a nutshell, our aim is to clone a piece of template DNA in a plasmid and then make RNA from that template. If you do that, you have an RNA expression vector. Let's talk about some general principles and considerations when working with RNA expression vectors. First, the usage. Why do you want to do this? Or what is your application? This will often determine the level of RNA output or the expression level. On a relative scale, this could be high, medium, or low. The expression level oftentimes determines the cellular stress. Second, you should know the host in which the RNA is going to be expressed. The choice ranges from prokaryotes to eukaryotes. The third consideration is around design and the choice of molecular components. Of course, first you need to know the template DNA which you desire to get the RNA from. This template will inform you about the size and complexity of both the DNA and the expected RNA. Then you have to think about the vector. What is its size, its copy number, its stability? Typically, an expression vector has all the features of a normal plasmid, but in addition, it'll contain promoter and terminator elements to cause transcription. Sometimes, you may have control elements in an expression vector as well. Now, let's understand how all these considerations are connected. If you were interested in high RNA levels, you would need a strong promoter. This idea is called the strength of the promoter. Conversely, if you needed low RNA levels, then you need a weak strength promoter. Often, control regions can be used to modulate the strength of a promoter. Our host preference determines the choice of promoter, terminator, and its corresponding control elements. This simply means that the promoter and terminator in one organism, say bacteria, may not be compatible with another organism. Likewise, promoters from animals may not work in plants. Our DNA of interest will change the size of our final vector. This oftentimes can change the copy number of the plasmid and its stability. Complex template sequences will also affect the stability of our final vector. These are some ways to think about design. Now here's a question for you. Given this information, what do you think will cause a cellular burden to increase, and why do you think it'll happen? Just a small note as we go forward in this video, I will not discuss control regions. We will come back to them in protein expression vectors. Our focus primarily is on promoters and terminators. Let me give a quick summary of some commonly used promoters and terminators in RNA expression vectors. Now, this applies to protein expression vectors as well. If you are expressing RNA in bacteria, typically promoters from lac operon, arabinose operon, or tryptophan operon are commonly used. You can also use viral promoters like T7, sp6, or T3 in bacteria. In bacteria, there are two ways to terminate transcription. One way is through extrinsic termination, which uses external factors like Rho, MFD, and Nus. Second is intrinsic termination, which depends on secondary structures like hairpins, which are formed by the RNA. If you use viral promoters, then specific viral terminators will need to be used. Yeast promoters are typically derived from genes like Gal4, alcohol dehydrogenase, uracil synthase, and many more. The most common terminator in yeast is from aldehyde oxidase and the cytochrome gene. People have also developed synthetic chimeras of these two, which may be more efficient in termination. Expression in humans typically involves using promoters from genes like phosphoglycerate kinase from glycolysis, the elongation factor 1 alpha from translation, the U6 from spliceosome, or some weird ones like CAG. Viral promoters work pretty nicely in humans as well. Termination sequence is used from the globin gene, typically ones from rabbit or rat, or growth hormone genes. A stretch of poly-A can also terminate transcription, although it's not as efficient. Plant-specific promoters often come from cauliflower mosaic virus, the opine genes, or the ubiquitin genes. The terminators often come from the same genes as well, among a few others. These are commonly used promoters and terminator elements. Do not take this as an exhaustive list. Let's try to work through an example design of an expression vector, and say our aim is to make RNA probes using just one template. The cloning will be done in bacteria. Broadly speaking, the aim is to produce and purify RNA probes, and we need them in large quantities. Through this exercise, we will see that the RNA expression in bacteria in this specific example is not a good idea. As an alternative, in vitro transcription is a much smarter way to get things done. So let's start. Case 1 is when you try to produce the RNA in bacteria. This is not a common practice, especially if you want to purify your specific type of RNA. But this is a learning exercise, and let's see how you would do it. In the vector, you will need 3 molecular parts. The bacterial promoter, because the expression will occur in bacteria. Then you need a bacterial transcription terminator. Between these two sits your template DNA. Using restriction enzymes or topocloning or your favorite cloning method, you can assemble this vector. I have already talked about the choices of promoters and terminators in bacteria. Once this vector is inside a bacteria, the bacterial RNA polymerase can recognize this promoter. If you were using lac-operon promoter, you can feed the bacteria lactose, and you will make a lot of RNA from this vector. Given a terminator, the polymerase will stop transcription at a specific point. This production cycle will perpetually continue inside a bacteria. Remember I said expressing RNA in bacteria for the aim of purification is a bad idea. Here's why. Note that in a bacteria, you've got both your plasmid and the genomic DNA. The bacterial RNA polymerase will bind on promoters from both these DNA, which means you make RNA from both plasmid and the genomic DNA. So our problem is that the RNA from our template is mixed with the RNA from the genome. Since we want to purify the RNA, we have to do additional steps to purify, which may not be a cost-effective strategy. So case 1 is not common practice for this particular situation. The alternative is to do in vitro transcription, also called cell-free transcription. The vector will look exactly like before, except that the promoters and terminators are different. And most commonly, viral components from T7 or sp6 are used. They tend to be a lot more compact. The terminator is always a hairpin type terminator, so it does not depend on any external protein factors. The assembly can be done pretty much the same way. Since viral promoter and terminators are so short, you can actually add them as primer overhangs and clone them scarless. The promoter recruits an RNA polymerase, but unfortunately, viral promoters are not compatible with bacterial RNA polymerase. Now, let's take a small detour and imagine you do this the uncommon way, which means if you wanted to express the RNA in a bacteria, you will have to engineer the bacteria to contain genes that make viral RNA polymerase. The viral RNA polymerase binds only to the viral promoter and makes RNA from the template. In this bacteria, the bacterial RNA polymerase is still active, making RNA from the genomic DNA. So even in this situation, you have this contaminating RNA. So what do we do instead? The solution to the problem is that you extract plasmid from the bacteria, typically one that does not express viral polymerase. Second, you extract crude protein lysate from a bacteria that expresses a viral RNA polymerase, and you add some RNTPs if necessary. So the bacteria is simply used to mass produce the vector. By the way, companies sell these things as ready-to-use enzyme mixes as well. Once you have these two things at hand, you take the plasmid and mix it with the viral polymerase in a tube. The viral RNA polymerase binds to the promoter on the plasmid and makes RNA. When the reaction is done, you simply purify the RNA. In this, there is no contaminating RNA from the bacterial genome. Since all of this is done in a tube, this is called in vitro or cell-free transcription. Here is some bonus information. Often, people don't like to add any Terminator sequences to their expression vector for in vitro transcription. Instead, you take the cloning site, which let's say can be cut with the enzyme C. You cut and linearize the vector and add that to the reaction. In this case, the polymerase will bind and make RNA from the template. But when it comes to the end, it'll simply fall off the template. This ensures exact termination of the transcription because sometimes in a plasmid, the polymerase can continue beyond the transcription termination point, and that will result in probes longer than the template, which is not good. This example of production and purification is not limited just to RNA probes. Hairpin RNAs for RNA interference and even mRNA vaccines are made using this exact same process of in vitro transcription. Let's do another example. This time, you're trying to express an RNA inside a eukaryote, say a human cell. This could be any cell, but let's limit ourselves to a human cell. Our assumption is that the RNA is non-coding in function, and you may want to do this for RNA tracking studies or making a guide RNA for your plasmid-based CRISPR experiment. The cloning of our vector is still done in bacteria. Same as before, our vector needs to have a human-specific promoter and a human-specific terminator flanking the template of interest. And likewise, you can assemble this with restriction enzymes or any other cloning method. We have already discussed promoters and terminators for humans that range from human or virus-derived. In the previous example, the viral promoter was not recognized by the bacteria. Likewise, when you clone this vector in bacteria, the bacterial RNA polymerase will not recognize the human-specific promoter. Essentially, the point here is similar. You're using bacteria to mass produce your expression vector. Once you have the vector ready, you transfer it to human cells, and that makes RNA from the template, because the human RNA polymerase will recognize the human-specific promoter. In plasmid-based CRISPR, the template often gives you the guide RNA, which then forms a complex with the Cas9 enzyme. We discussed the RNA part of it. The other task involves producing a protein. We will see it in the next video on protein expression vectors.